Our final lecture on antimicrobials will deal with antiviral drugs, with a particular focus on HIV and AIDS. So viruses are small infective agents consisting of nucleic acid, either RNA or DNA, enclosed in a protein coat as shown in the diagram on the right. They are not cells and utilise the metabolic processes of the host cell that they infect in order to replicate. DNA viruses usually enter the cell nucleus and direct the generation of new viruses. RNA viruses direct generation of new viruses without involving the host cell nucleus. RNA retroviruses possess an enzyme called reverse transcriptase, which makes a DNA copy of the viral RNA, which is then integrated into the host cell genome and directs the generation of new virus particles. This slide shows some of the most common viral infections and the anatomical locations where they tend to affect, including the common cold, pneumonia, STIs, and skin infections. The human immunodeficiency virus, or HIV, is an RNA retrovirus with two major kinds. The HIV-1 is responsible for human AIDS, and HIV-2 is largely confined to Africa and is less virulent. Interaction of HIV with the host immune system is complex. It is known to infect T cells, particularly CD4 positive T cells. The cytotoxic T lymphocytes direct the killing of virally infected cells and produce and release antiviral cytokines, while the CD4 positive T helper cells have a direct role in the control of HIV replication. HIV is incorporated with the host DNA and can remain latent for a prolonged period of time. Mutations in the HIV genome enable the HIV virus to escape recognition by the original cytotoxic lymphocytes, requiring the production of new lymphocytes that ultimately can exhaust the immune system. The diagram on the slide shows the production and role of T helper cells. As you can see on the left, the antigen presenting cell, or APC, presents antigen in a major histocompatibility complex, which is essentially an endogenous protein that combines then with the antigen from the virus or the microbe, whatever it might be, presents it to the CD4 T cell, which can then decide which type of response is going to happen in the effector phase. This could be an antibody-mediated response or a cell-mediated response. If it's an antibody-mediated response, it has to interact with B cells, which produce B cell receptors initially that ultimately, when they're mature, start releasing antibodies when they form plasma cells. These antibodies are made to specific part of the antigen and can then recognize or tag cells that contain those particular antigens on their surface. If it's a cell-mediated response, it will train cytotoxic T lymphocytes to kill virally infected cells. The HIV virus actually attacks the CD4 positive T cells, and therefore the T helper response is effectively removed from its ability to train the adaptive immune system. The diagram on the left shows the life cycle of the HIV virus, which can target T helper cells and that are central to the adaptive immune response. It is important to have good knowledge of this to understand where we could pharmacologically target it. The HIV virus fuses to the host cell surface via an interaction between GP120 viral protein and the CD4 protein on the host cell. From that point, viruses can insert their nucleic acid and proteins, including key enzymes, into the cell. The next step is to turn its RNA into DNA by the enzyme reverse transcriptase. This viral DNA is then transported across the nucleus and integrates into the host DNA. New viral RNA can be made in the nucleus during transcription, and it is then used to make new viral proteins that move to the cell surface and form new viral capsids and mature into new virus that can enter a new cell. The graph on the right shows the effect of plasma viral levels, or titers, shown in green, on the number of CD4 positive T helper cells, shown in yellow, over time. It also shows the appearance of symptoms over time and even a period of clinical latency where the virus can remain hidden for many years. Finally, what patients die from is usually an opportunistic infection that could normally be dealt with if they had enough T cells, T helper cells. The goal of antiretroviral therapy is to achieve and maintain suppression of plasma viremia to below the assays level of detection. It also aims to improve overall immune function as demonstrated by increases in CD4 positive T cell count. It aims to prolong survival, reduce HIV-associated morbidity, and improve overall quality of life. Finally, it aims to reduce risk of transmission of HIV to others. Current antiretroviral regimens do not eradicate HIV. Viral rebound can occur rapidly after treatment discontinuation, and this can often be followed by CD4 positive T cell decline with a potential for disease progression. Strict adherence to prescribed regimen is essential, therefore, in order to avoid viral rebound and the potential for selection of drug-resistant mutations. A combination regimen should consist of preferably 3 but at least 2 active agents based on genotype-resistant test results. Selectivity for viruses is difficult as they reside within the host cell. Some enzymes are virus-specific and thus are useful drug targets. Most antivirals are effective only when the viruses are replicating. Antivirals are grouped according to their mechanisms of action and side effects. Shown in the diagram on the right are the points in the HIV life cycle where current drugs can target. These include chemokine receptor antagonists, inhibitors of viral fusion, reverse transcriptase inhibitors, viral integrase inhibitors, and protease inhibitors. The first class we will discuss is the nucleoside reverse transcriptase inhibitors, as shown in the diagram on the right. Nucleoside analogues are phosphorylated by host cell enzymes to give the 5' triphosphate version. This then competes with the host cell triphosphates essential for proviral DNA production, thus causing DNA chain termination. An example is zidovudine, which is a thymidine analogue, whilst newer NRTIs such as abacavir have less toxicity. The non-nucleoside reverse transcriptase inhibitors do not require phosphorylation. Instead, they bind to the reverse transcriptase, causing a conformational change, reducing its activity. An example includes nevirapine. Most drugs in this family are potent inducers of cytochrome P450, which is an important consideration if other drugs are to be used in combination. As shown in the diagram on the right, protease inhibitors bind to HIV proteases, blocking the proteolytic activity. This results in the provirus being translated into 2 biochemically inert polyproteins. Protease inhibitors prevent the cleavage of the nascent viral protein into functional and structural proteins. An example of one such drug is ritonavir. There is often a low likelihood of resistance with this drug, even though it is a potent inhibitor of cytochrome P450 3A4. On a side note, DNA polymerase inhibitors can be used as antivirals as they target a key enzyme involved in DNA replication. Aciclovir is the prototype drug. It is a guanosine derivative, as can be seen from its structure on the top right. It is converted to the monophosphate form by viral thymidine kinase, as shown in the pathway in the bottom right. It is 30 times more effective against the herpes virus enzyme than the host enzyme. Following the viral thymidine kinase action, host cell kinases then convert the monophosphate to the triphosphate form, which inhibits the viral DNA polymerase enzyme and terminates the nucleotide chain. Resistance occurs by changes in the viral genes coding for thymidine kinase or even DNA polymerase itself. Integrase inhibitors act by inhibiting HIV DNA integrase, as is highlighted in the diagram on the right. This prevents the formation of covalent bonds between the host and viral DNA. It prevents the incorporation of viral DNA into the host genome when forming the provirus. An example is ratelgravir. It is used in the treatment of HIV as part of a combination therapy due to its potency and low potential for side effects and drug interactions. Inhibitors of viral entry and fusion include the chemokine receptor antagonists and the fusion inhibitors. Both are highlighted in the diagram on the right. CCR5 together with CXCR4 are cell surface chemokine receptors that are exploited by some HIV strains to gain entry into the cell. An example of a drug used to target this is mariveric, a chemokine receptor antagonist that is used in combination with other drugs. Fusion inhibitors prevent the conformational changes necessary for the fusion of the viral and cellular membrane. An example of a drug used here is enfuvirtide. Its novel mechanism means no cross-resistance with other antiretrovirals, but it needs to be administered parenterally. Other strategies to destroy virus includes using monoclonal antibodies and immunomodulators that modify immune responses, such as antiviral molecules like interferon. Monoclonals are immunoglobulins, essentially, that are antibodies raised against epitopes, potentially on viruses. Pavlizumab is a monoclonal antibody directed against a glycoprotein on the surface of respiratory syncytial virus, or RSV, and is one of the first monoclonal antibodies we have against such a glycoprotein. Other antibodies are now being tested for such viruses like HIV. Interferons are a family of inducible proteins, with alpha, beta and gamma forms being the most commonly found members. Interferons bind to specific receptors on host cell membranes that then induce the production of enzymes that can inhibit the translation of viral mRNA, as is shown on the diagram on the right. The result of these enzymes is that they inhibit the translation of viral mRNA into viral proteins, thus stopping viral replication. These can be made recombinantly in the lab, and they need to be administered intravenously, but they do not usually cross the blood-brain barrier. It has been known that combination therapy involving different agents targeting different steps in the life cycle of the HIV virus results in synergistic or additive antiviral effects. The results can be seen on the graphs on the right. These show both a reduction in plasma HIV RNA and an increase in CD4 T cell count and T cell activation over many years. HART, or highly active antiretroviral therapy, typically consists of 2 nucleoside reverse transcriptase inhibitors with either a non-nucleoside reverse transcriptase inhibitor or 1 to 2 protease inhibitors. Other drugs such as the HIV integrase inhibitor Aciclovir, the chemokine receptor antagonist mariveric, and the HIV fusion inhibitor infuvritide may also be used in combination therapy. Some patients may experience treatment failure and require more complex therapy. In recent years, the use of pre-exposure prophylaxis or PrEP has been trialled in certain at-risk groups.